Thank you, Erica. Thank you, Erica. Thanks for the introduction, and thanks for everyone for joining here today for the next 1 hour. I'm gonna be talking about what eDNA task group, what we've been doing, and what kind of goals we have, and give you some updates on our activities. So I'm gonna share my screen. Hang on. All right. Can everyone see the main presentation?
That looks good. Yeah.
Great. Okay, cool. So, all right, nice to meet you, everyone. My name is Miwa. I am a research scientist at CSIRO based in Australia. So today, I'm a convener of this task group. So I'm going to give a overall activities that are what we've been doing for the last 1 year, and then what we are going to do in the next year or so. So throughout this, yeah, I'm gonna give you those updates, and then at the end, I'll be happy to receive any questions you may have, and also any feedbacks or suggestions on our approach or strategies. Those things would be really valuable as well for us to hear. Yeah, so there will be some time for that at the end. Cool. So who we are, this is the web page about the task group. So my name is on the top as a convener, and then we have quite few core members, and all of us are working really hard to achieve some of the goals around eDNA data. And so I am from CSIRO, and then I also lead the project initiative called Making eDNA Data Fair, or Fair eDNA. Now we call it FAIRY initiative. And then many of the members are amazing eDNA researchers from various institutions or universities or industries. And we also have people from those biodiversity databases, GB Phenobis, I'm sure you're quite familiar with, and some molecular data specific database such as eDNA, and GSC is a Genomic Standard Consortium. And many of us are also connected through different type of networks and task groups. And for example, Auburn is one of them. Some of us are from eDNA Aquaplan in Europe. Also, ISTIF is an international eDNA standard task force. Some of us are member of ISTIF. And so, as you can see, we are from various backgrounds. Some are from governments or universities or databases, standards, but we are all like one family, I guess. And then we're all connected and we do a lot of things together. We also have extended families like Ovon and Eden Aquaplan. And then, so we have a really amazing collaborative network here. And I should also mention that we have another task group and the Genomic Standard Consortium, eDNA Working Group, but basically the members are identical. I'm leading that and we have the same members. So we created these two task groups because we are working for Genomic Standard Consortium. We are trying to create eDNA checklist for GSC. And then also third week, we have a similar goals. So we have a separate task groups under different organizations, but our objectives have a lot of overlaps. And then, so we're working together parallelly, concurrently, and at the same time towards the same kind of goals. So eDNA data or metadata challenges, I listed a few here. One of them is workflows are highly diverse. There are many different workflows, starting from a sample types can be different, and then multiple step sample collection, DNA extraction, bioinformatics. All these things can be very different depending on the environment or the target taxa we're looking at or resources we have all use cases as well. So that's the second part, high diversity of use cases. And depending on what we're using eDNA for, the stringency or like the accuracy that we need for species assignment can be also quite different. And because that is different, then we have a different workflow or different strategies towards our goals. And then, so there are uncertainty in species assignment. We acknowledge that can be one of the limitations in eDNA. And those uncertainties come from different components. And then we do our best to tackle those uncertainties, but then how we tackle them can be different depending on various factors. Then there are quite complex relationships within and between samples and sequence data. So I guess some of those challenges are quite common for any kind of biodiversity studies, not specifically for eDNA, but some of them are eDNA specific. And then so we're tackling those things by developing a metadata checklist, a comprehensive metadata checklist for Tadwig communities. So for that biodiversity context to address these challenges. And how are we going to do this? So we're reviewing an existing checklist that are relevant for eDNA, and we want to propose new terms to Tadwig that are still missing in the Darwin core context. And so yes, so the fourth one here, complex relationship between samples and sequences. We acknowledge these kind of challenges in the eDNA data, but I think this Darwin Core Data Package team is doing amazing work to tackle this. So it's not just for eDNA part, but in many of us see this complexity is quite challenging to address through metadata format. So I'm hoping that they can solve most of the challenges that we have, and so I would love to chat about it with them. And I think it's already happening through many tables that they created and how they link the tables. This has been really amazing and really useful for us. So I wanted to highlight that part. So I mentioned about existing metadata checklists for eDNA. And so what are out there now? There are quite few, for example, mixes, Darwin Core, you are probably all familiar with those, and GGBN, Global Genome Biodiversity Network, and minimum information from qPCR experiment, Mike or Mike, I'm not sure how to pronounce this. So there are quite few out there. And then a few years ago, GBIF has developed these DNA-derived data extensions, which are covering the majority of those one. And then I started this FAIR eDNA or FAIRY project almost 4 years ago. And through that, we developed a FAIRY guidelines. And also, so in the guidelines, we talk about how to format the data, what kind of data components are important in the eDNA context, but also we developed a very comprehensive FAIRY checklist, metadata checklist. And then so we review those existing metadata, and then we identify some gaps in the current checklist. So we created quite a few terms, and then that is the result of this FAIRY checklist. So we built upon those existing checklists, and then we built more on top of it. And so that is the overall of existing checklist. I would say this FAIRY checklist is probably the most comprehensive checklist for eDNA workflows to date. And then after this FAIRY checklist was developed, then Darwin Core Data Package has done a lot of progress, and then they now have molecular protocol, nucleotide analysis, those kind of tables that are very relevant for eDNA workflows, and there are quite few terms within those tables that are coming from Darwin core and DNA-derived data extension. So that is really useful for us as well. But we also have quite a lot of other terms that we created. So we want to propose those terms to Darwin core or maybe data package teams. And when I mentioned like that fairy checklist is very comprehensive, we have about 330 terms, and then half of them were newly created, but the other half came from those existing checklists or other kind of standards that are also listed here. And quite few come from mixes, but then others are already in, these guys are already in Darwin core data package. So these terms are, for example, in the mixes, but also in DNA-derived data extension. Then the Darwin core data package team took all the terms from DNA-derived data extensions. So they're already in the data core, Darwin core. And some of them are really like just the original Darwin core terms. So those terms, we don't need to worry about them. But what we are thinking to propose to Darwin cores are mostly from the other components. And the examples, so there are 158 terms here that are newly created by a FAIRY working group. And just to give you a few tiny handful examples, for example, inhibition check, how the PCR inhibitions were monitored, and was the blocking primary used, and then if it is, what are the sequences? Second, PCR protocols. So first PCR protocols are quite well described using mixes terms. But then in eDNA metabarcoding workflows, we often do the second PCR, and then that part cannot be described using those current existing terms. So we created quite a few terms related to the second PCR. So there are quite a few things like that. And then, so these are the terms that we are reviewing at this moment and thinking, shall we propose to the Tadwig? Shall we also propose it to GSC? So we call that FAIRY term review. And with quite time-consuming work, we are reviewing term by term for all these 330 plus terms. And we have a GitHub page. And in the FAIRY GitHub page, there's a repository specifically for this term review. And we created issues, one issue per term. And in each term, we check, is this term already in the Tadwig or Darwin core? Is this term already in the mixes? If not, shall we propose to these two organizations? So this is what we've been doing for the last few months or so, spending quite a bit of time on it. So I just give you one example here. There's a term called filter passive active. So was the filtration, like for example, water or air samples filtration, has it been done actively or passively? And then if you click this issue, then there are a few comments that I add here, and they, how can I say? So for each Darwin core and the mixes standards, we review. So yeah, sorry. So this is the close-up of these trees because I guess the original one is too small. But if you look closely, so this is the filter passive active, that's the term name. And does this exist in Darwin core? And no, it doesn't. Does it exist in another standard like mixes? No, it doesn't. It's a new FAIRY term. And shall we propose it this term? To Tadwig, then yes, we should. Is it a controlled vocabulary? We look carefully about this controlled vocabulary because we want to make sure the entries are as machine readable as possible. So wherever we can make the term into controlled vocabulary, we did that. So if that's the case, then we also want to propose that as a controlled vocabulary. But in this case, it's a Boolean term, 1 and 0, but it is not really a controlled vocabulary. So we said no. And then, okay, let's propose this, put that into the proposed term list. So this is all this kind of like decisions and the criteria that we're looking at, and then we make a final call. If we have any comments, then we also add comments here during the discussion, or also not only during the meeting, but also offline. We sometimes review that and people can comment. Anyone can comment here. So if any of you are interested in looking into some specific terms or overall thing, please go into those GitHub page and then put any comments, and anything will be quite valuable for us. So we appreciate that. And then once we make a call, then we put this kind of labels. So at the end, we can have a list of the terms we want to propose to Hardwick or GSC. So most cases are quite straightforward like this. If it's not in the Darwin core at this moment, but Fairy created, for example, or it's in a mixes now and it's relevant for eDNA, we want to propose that because during the development of Fairy checklist, we Fairy eDNA community thought it is a useful term in eDNA metadata. So that's most of the case, but sometimes it's quite Fairy specific term, like a Fairy checklist version number. Things like that is too specific for Fairy framework, not so much for the Hardwick general community. So those terms, we don't include that into our proposal. So we're doing that kind of review term by term, and repeating the same thing for mixes. And here I want to show, I'm not sure if Florian is in the audience. In one of his email he sent to me and other people, he is a co-chair of ISTIF, International eDNA Standardization Task Force, and he mentioned he wrote that this, and I found that it's really relevant for us. We are a task force doing lots of boring, time-consuming, but extremely important work. And when I read this, it made me feel good about what we're doing because it was exactly how I felt. Lots of boring. Well, sometimes it can be exciting. Some discussions can be exciting, but can go quite boring and repetitive, very time consuming always, but extremely important work. That's what I would like to believe. And I think it is. It is. So it was nice to hear these words from Florian and then reassure what we're doing is right. So this is what we've been doing, spending quite a bit of time in the last few months. So that was about the Fairy term review. But in overall activities for the last 1 year, what we've been doing is the online fortnightly meetings. So we're meeting quite regularly every 2 weeks. And we also had a chance to have an amazing in-person meeting in UK, which is very unique and exciting because we always meet online. And so there was a GSC 25 meeting in the UK, and quite a few of us were attending anyways. So I decided to run a workshop, like task group meeting, and then we applied this community support funds, which could bring a few extra people to UK. Without these fundings, those people couldn't travel to UK. So that was a huge value for us to bring those people, and we're really grateful and thankful for this Hardwick support. We run 2 days meetings specifically for eDNA. Well, one day it was quite big group with the age and DNA, eDNA group, and then NCBI, quite a few GSC people joined as well. So it was really, really informative and useful for us, but also another day of like really specific to this task group. And then we discuss a lot about this Hardwick task group goals and activities. Then we spent 3 days together for the conference. And then another main activities is what I mentioned, the Fairy term review. So we did quite a bit in those fortnightly meetings, but each meeting is 1 hour and we don't get much done within an hour. So we decided to do a 3-hour hackathon, really focusing on those term review. And we've done 2 of them, and that was really productive. Yeah, so we're gonna keep on going until we finish with all the theory terms. We've done about a third of them, about 100 terms. We're getting quicker and better, so I think we can smash the rest. Yeah, with less time, I think. And so apart from, well, before we started this term review process, what we discussed in those in-person meetings and then fortnightly meetings is like, for example, how do we develop eDNA metadata checklist that meets Tadwick and GSC scopes and needs? And so it was quite a bit of like discussion, trial and errors until we got to that strategy that I just showed you. So we have those GitHub repo and the issue, and each issue is a term. We have these decision trees and then put these labels. Now we have these systems and it's up and running. But until we get there were quite a few discussions and then trial and error. So that's what we did last year. And then we also discussed how do we map these Darwin core mixes and varied checklist. So we've been talking about it. We learned about triple SOM. We saw David from Mapping Working Group. He just ran a session a few hours ago, so I joined that one. He also invited me to their working group meeting. So it's been really, really useful for us to learn about how to map and then how, what kind of support we could get from them in the future once we actually start mapping. So I'm really grateful with the support. Some of us from the working group is also connected with the peer and then Raisa Mayer from the Darwin Core Mixes Interoperability Task Group. So I'm sure we can get some support from them. So we haven't really started the actual mapping process yet. But once we start, I'm sure we'll come across quite a few challenges and questions, and I'm hoping that we can get some help. So from those people. Then we also discussed, especially during this in-person meeting, spent quite a few hours on how to define a species assignment confidence level. And I want to show a few slides in the next after this about this confidence level. The main reason is because this was exactly 1 year ago during this annual task group session. I presented about about the eDNA task group, and then there were a couple of comments from the audience about this. It'd be good to have this kind of confidence level score or some sort because that could be useful. And so I identified that as it's a demand or niche or needs from the communities to sort this out. So, and I thought we could try and work on that. So we did try, and I want to show what we discussed, what kind of attempts we made, and the conclusion we came to. So in the eDNA, species assignment can be quite complex. So we rely on DNA reference database. So we get the eDNA sequences and we compare that, we match that with reference database from a known species. Then we know what species that was in that sample. But it can be 100% match, then we can be quite sure that it was this species that was in that location or in that sample. But then sometimes it's a 97% match. A few bases are not matching exactly. And that could be because of the sequencing error or because of the just little tiny variation between a population within a species. Or it could be that that species of the eDNA sequence coming from is not in the reference database. So there are various sources of errors that can make the classification not accurate or at lower taxonomic resolution. So instead of at species level, we can only identify the genus level. So that can happen. And then we can also have a primer bias during the PCR. eDNA samples are very highly susceptible to contaminations, like cross-sample contaminations or human or lab contaminations, because we are working on the tiny, tiny trace amount of DNA. So that can happen. Then we do have lots of screening systems through bioinformatics, but then that kind of stringency in the bioinformatics, what we apply can be different depending on the purpose of the studies. And then all this, what I explained now could be quite complicated. And then there are various workflows and how to understand for non-eDNA users. So we all acknowledge all these kind of challenges. During this in-person meeting, we discussed a lot about it. How can we do this? Can we make some sort of like index or score for non-eDNA people to use? Even for eDNA people, that would be really useful to have. So we come across to this paper. The title is Confidence Scoring Procedure for eDNA Metabarcoding Records and its Application on Global Marine Fish Datasets. So it's specifically for marine fish. And they put this kind of criteria, like 5 criteria to define the score. One is that does the species identify in eDNA samples? Does it actually occur in that area? Yes and no. And then what is the percentage match to the reference database? I mentioned about like 100% match, then that can be, quite assuring. Then it's a score 1, but then if it's lower, then it's a score 0. And so there are several criteria, and at the end, they just simply add up this number, and then you have the scale from 0 to 5. And I thought that sounds great, and then that'll be amazing if we can apply that. But we're talking about a global database from not just fish, but so many different taxa in GBIF and OBIS. And we came to this conclusion that this could work for fish because fish is quite well-studied taxa, and we know what species are in that area. We know if the reference database is complete or how much is missing. We can kind of figure that out. But for many other taxa, like invertebrates and fungi, and that's not possible to do that taxa by taxa and then region by region as well in this case. And then there are so many other factors to consider to define the confidence level. So we asked the AI, Gemini, gave a list of fairy terms and then asked Gemini to identify which one could be used to define the confidence level. And there were so many. I'm not sure I can't remember if those are all of them, but there were a lot. Detection criteria, percentage match, and lots of error rate cutoff, all this kind of like thresholds for bioinformatic pipelines. So it's very, very, very difficult to make some sort of model to include all these kind of things and then define the confidence level. So at this point, we came to the conclusion that this is not a priority task as a task group. And what we should be focusing on is to define this checklist. So if we make comprehensive metadata checklist, including all of this, then at least we have this information in a machine readable way in every eDNA metadata. Then in the future, if we identify, okay, this is definitely what we need, and I'm sure there's not only one, but multiple, to define the confidence level, then we can extract those information and then we can make some sort of like modeling or algorithm to define the confidence level. But at this stage, it's a really big task. And then we really wanted to focus on the checklist establishment first. And then maybe we move on to this task, or we might work with GBIF and OBIS and then work together on this, because I guess this species assignment index, confidence level index, is not only eDNA specific, but I'm sure it happens to many other kind of species assignments. So maybe we can work together with them. So this is something that I wanted to show about species confidence level. because it was raised in the last year's session, and then I think some of you might be interested in, so I wanted to show that. Okay, so what we are planning to do in the next year or so, or more about maybe immediate next 6 months or so, the rest of 2026. So I mentioned multiple times, we are going to complete this fairy time review in this fortnightly meeting, so online hackathons, and we aim to complete by September this year. And then once that's done, then we're going to map those terms. Then once the mapping is done, the next step is to actually make a mapping tools based on this mapping that we did. There are already quite a few tools for fairy formats, so it's to change the format and the fairy terms into database specific required formats and terms. So I guess these are Darwin core, and then these are mixes terms, and then formats are slightly different. So there are multiple tools already there. It's all available in the GitHub. And well, meta barcoding toolkit of the GBIF. Thomas just implemented this fairy format as one of the input format options, so which is great news for us. But it's still in the testing stage, and so it's not up and running yet, but it is coming soon. But once we make this, updated Darwin core mixes, new checklist with the proposed terms, the terms that we are proposing, then we need to update these tools after that. And then in August, I'll be attending a GSC 26 meeting, and then we just got notification from GSC that I'm receiving this Dawn Field Award this year. So I guess this award is really going to everyone in the task group, not just me. It's the whole group achievement, and it's nice to be recognized and the importance of our work like that. So yeah, so that's one big achievement we got. And then in October, we are planning to have an in-person meeting in Seattle. So there is an eDNA conference that many of us will be attending. We'll be all traveling, or not all of us, but it's the same concept of how we run in-person meeting last year in UK. Few of us are going there anyway for a conference, so why not we make a day or two of a task group meeting? So that's what we're planning to do. And in the first day, I really want to focus on this task group meeting, and we want to finalize this term proposal to third week. So should be mostly done by September. So this October will line up really well in time wise. Then also the mapping part, we want to complete that. Or if there are any big challenges, then it will be a good opportunity to discuss on that day, because this kind of in-person meeting, not just 1 hour online meeting, but we have a half day or full day to discuss in the same room together. It's a huge difference. Like we really felt that last year in UK. So yeah, I think it's going to be really effective to have this day. And then we also have a workshop planned. This is for eDNA practitioners. And we're gonna introduce about this kind of tools, so MDT and then the fairy, and then yeah, so it's gonna be another day, and then we have a conference together. And we're planning to apply this CSF again this year for us to bring few more members. So yeah, 6 members are confirmed to go there, and we have fundings from our institutions. But then a few of us would like to come, but then don't have money to go. So this CSF would help massively to bring those few extra members. And yeah, so we will send applications soon, and please consider about us, because we're going to be achieving these things when we meet together. Okay, thank you very much. I spoke quite a lot for half an hour. I hope it wasn't too much for you. But yeah, I would like to thank all these task group members many hours of meetings together, bringing all these brains together, and it's been really amazing to have everyone in that team. We are working very hard towards our goal, and then all the Tadwick supports, of course, and also Cecilia and Thomas from GBIF. They are not in the task group, but almost the task group members, I guess. Yeah, I contact them a lot and get their inputs all the time. So really appreciate that. And then also the original Fairy members and many more. So that's it from me. And now I would like to ask the audience if you have any questions to me, or as I mentioned at the start of my talk, I would appreciate any feedback or suggestions. Like if, we're quite well connected with different networks within the eDNA space, but if you think of like another task group or activities that are happening elsewhere, and then maybe we can connect or exchange some information, I might be missing out some of those groups. So if you have any suggestion like that, that could be really useful. And yeah, anything basically. So please ask any questions.
Mila, there's been some questions and discussion in the chat too, so you might want to just take a second, catch up on that.
Okay. Might need
And
more than a second.
folks who ask questions in the chat, you might want to unmute yourself and ask them verbally now instead.
Steve is there. Yeah. Cecilia, are there terms you're considering proposing to both Tadwick and Mixes? Oops. Sorry, someone posted another one and it took me away. Looking at the labels for the issues, it seems that you may want to propose both standards, but I wonder if you can give an example of such term issue and why it would need to be proposed to both standards. I guess I'm not sure if I understand your question properly, Cecilia, but
I can. Sorry, is it okay? It's because when you showed the GitHub repository of the issue tracker you have, I saw there were some labels that both said proposed to Mixes and proposed to Tadwick. So I was just wondering if there are terms from you come up with during your ferry work that you are considering proposing both to Mixes and to Darwin Core or to Tadwick. Because then I would like to know like an example of why it would need to be in both standards, the same term.
Because I guess, GSC maintains and organizes mixes, and not that like it's not like all mixes terms are in Darwin Core, and vice versa as well. So some of the terms are really biodiversity specific stuff, and then it will be interest for Tadwig, but then GSC is only interested in something that is related to sequence data. So that won't be in the GSC. So there are slight difference in the scopes, and that's why we are reviewing both of them. And if we think it applies to both, then we were planning to propose to both. But do you think we should be just proposing to say GSC first, and then you would take it into Tadwick?
Yes. Yes, at least that's what we've done for Darwin Core data package. So if it's in Mixes, we can easily, I think there should just be one standard body governing the term moving forward instead of having duplicates. And we could easily, if it could be proposed to Mixes, I think it should be proposed to Mixes rather than Darwin Core. But of course, if there's terms that does not fit in Mixes, maybe it makes sense to have it in Darwin Core. But maybe Steve has a comment on this also.
Well, I think that maybe I'm wondering if I misunderstood how it worked, and that might be why we're doing it this way. So from what you're saying, Cecilia, that makes sense. I think it was just that we wanted to make sure that the term was included in the extension. And so perhaps it's that the extension isn't something that Tadwig oversees. And so maybe we're looking into the wrong space. Like we're not trying to create two separately managed terms. The idea would be that the term would be in one or the other standard, and then, like you said, ported to the opposite one.
Yeah,
Um,
I think that's doable. It's at least it just requires, I don't even know if it requires a ratification process,
yeah.
but yeah,
I guess with the DNA extension, technically that's not Tadwig ratified, right? That's a GBIF managed extension.
yes, that's nothing to do with, well,
Yeah.
yeah, yes.
But
Okay.
then it's going to be technically ratified once the Darwin Core data package implements those DNA drive extension terms.
Yeah,
That's what
but that
I'm
wasn't the
understanding.
terms as such. That was the conceptual model, and so the mixes terms, if you propose them to mixes, they could be incorporated as far as I understand, unless it has a change to the schemas and the conceptual model. I don't think it requires ratification. So what I'm trying to say is that if you have terms that could fit in mixes and could be proposed to mixes, then you don't need to propose them to Darwin Core. But if you have terms that do not fix in mixes because of the scope, then it makes sense to propose them to Darwin Core.
Okay. So, well, I guess it doesn't hurt to keep on going with our process of this, fairy term review and then labeling those things in the GitHub issues. But then if we identify, we think it's a scope for both GSC and Tadwig, then we might include that into GSC proposal. And once it's ratified by GSC and then it's in the mixes, then you can take it from there.
Yes, but it would be nice to have the overview. If just I'm thinking about how do we keep track of all the terms you're suggesting to mixes and make sure that if they're relevant to include in Darwin Core data package moving forward, for example. But we can discuss that on the site, maybe.
Well, I think, yeah, I think that what we have will already accomplish that, Cecilia. But I think just the difference now is maybe we just have to change our language a little bit to say, okay, it's not that we're looking for these to be ratified by Tadwig, but just to make sure they're included in the extension and therefore the sort of the body of knowledge of biodiversity publishing. Right, that we want to make sure that when people publish to GBIF, OBIS, and other places, that they know these terms are available and that it's recommended they be included.
Yeah, and we also need to find the place for them, I guess, in Darwin Core data packages. Is it in the molecular protocol table? Is it in, the material table? But that's the mapping exercise, I guess, also.
Yeah, and there will be terms that we don't propose to GSC, but we would like you to consider. For example, those qPCR-based detection, species detection. And I raise this example quite often because that was an obvious example that GSC would not be interested in because it doesn't come with the sequence data, whereas GSC is really about sequencing data. But it does provide a lot of, eDNA-based derived species occurrence record, which I hope GBIF and OBIS you will be interested in. So
I think
those
very
terms,
interested.
I guess we can mark as proposed to Tadwig, but we wouldn't be marking as proposed to GSC. So with those labelings, I guess we can easily identify which terms are specific to which standards or common to both standards, and we can make a list and provide it to you, show it to you once we have that. Hope that works. Okay, cool. Thank you for the comment, though. It's really useful. And I see James, completely agree with your conclusion on the confidence assessment based on my experience. Thank you. Yeah, maybe you had a similar experience as well. Yuta, how are you using LinkML in your daily work on the eDNA standards? What does it enable you to do?
I can answer that,
Steve.
Neva. Yes, I
Yeah, I was
put it in
gonna
the
throw it
chat,
over to you.
and I
You
think what
already
we've
answered. Thank you.
no, I did
Yeah.
answer, but I think what we've uncovered here maybe is a little cross-pollination that could be healthy between the GSC and the Tadwig communities. So LinkML came from the national, well, it came from a community of people, but the what is it, the National Microbial Data Center, something like this, the NMDC, which is a lot of the same people that are developing GSC standards. And for me, it's once I sort of understood the basics, I found it to be a really nice modeling language. And it comes with Python packages for turning that model into JSON, into HTML documentation you can publish through GitHub, etc. So yeah, I think it's just a useful tool to have that may or may not work well for some of the Tadwig work. So I'd be happy to come talk to the Lattimore Core folks and then whoever else too.
Good, thank you. And then Yuta mentioned that also she runs a monthly meeting. So yes, might be good for us to attend and introduce about our work. I'm sure Steve can explain a lot better than me, but we can both attend. And then Cecilia for mapping different standards. With Mixes version 7 and Darwin Code DPI will require certification of both, which is still pending. Great to work with your task group on this since you already have done a lot of work on this. Okay, yes. So yes, we are working with Mixes, and we're not going to be included in the next version of Mixes because they are about to release in a couple of months, and we're not going to make it. But for the next version 8, eDNA checklist should be there. So yeah, and then we definitely need to work on this Darwin Core maintenance and the memorandum framework for those two organizations. Okay. Clara. I was wondering if you plan to process the terms also to the European Nucleotide Archive. Sorry if you mentioned it. Yeah, that's good. So European Nuclear Archive, eDNA, yes, we've been working closely with them. Joanna and Peter from eDNA are in our task group. And so I guess like one of the main reason we want to work with the GSC, or we want to get the eDNA checklist ratified by GSC, is because once that passes, once our proposal passes through GSC, then eDNA and CBI, they all take Mixes checklist. So automatically through the process, eDNA will be taking those terms that we're going to propose. So that's the plan we have. And then Sophie, not a question, but some feedback as we've been using the FAIRY guidelines and the metadata checklist, among other resources like the OBIS and GGB guides, to start working on the best practice guideline aimed at eDNA data publishers in France. And it's been extremely useful to us. So thank you. Thank you for saying that. We're also very interested in new terms and in mapping. You've been working on the between standards with GB France team and the colleagues at MNHN. Okay. Yeah, thank you for the comments. That's very nice to hear. And then I guess you are in the GB France team, so I'm sure you're very well connected with Tobias and Cecilia, and so I'm sure we have a chance to meet as well. Cross-pollination, yes, thank you. All right, does anyone have any other comments? You don't need to type in the comments, so you can just speak out or raise your hand as well. Okay, so I guess I give it back to you, Erica. And yeah, I think that's all from us.
Yeah, thanks everyone for joining. Thank you, Miwa, for hosting. This session was recorded and we'll be posting the recording on the Tadwig YouTube. So you can look for that if you would like to follow up on anything. And I will set you all free. Thanks.
Thank you very much. Thank you, everyone, for coming.